So the methodology that people use for sequencing is more often and most cheaply is called sequencing by synthesis. So to begin, just imagine a small DNA molecule attached to a surface. And so you have DNA here from the cell, coming directly from the cell. And the first thing that you need to do is add a primer, which is nothing but a complementary shorter DNA fragment to the DNA that you are trying to read. So at the beginning of it, you just take a smaller DNA fragment that just finds its complement. And then the free nucleotides can be added by the polymerase once it has this initial fragment of DNA. So the polymerase will come, will find the free nucleotides, will start adding them as they are complementary to the chain, to the original chain. These nucleotides have a special feature, which is once you add one nucleotide, it stops. So the polymerase stops here, detaches, and then you wash everything away, and then you apply light. This light will make the nucleotide fluoresce of a certain color, which was labeled before the whole process started. And that can be captured with a picture. So you apply the light, take a picture, and there you have a dot with a color. And then you go back to step 2 and repeat, and repeat, and repeat. For different nucleotides, each one of them labeled with a different color. And so what you have at the end is, from your initial DNA sequence, that is a single strand, you can, by adding nucleotides one at a time, applying light, taking a picture, you can start knowing how this original strand looked like. And so here's where one of the errors, the first errors of the process comes into play. So because the polymerase was taken from the natural environment of the cell, it's a living machine and it makes errors. And those mistakes are simply, it sees some nucleotide and adds a different one than the one it should be adding. And that might sound bad in this context, but let's remember that those errors are the main driver of evolution in real life, because those errors are mutations that are going to prompt evolution in the long run. But for here, we consider them errors. So the nucleotides get added, the pictures get taken, and we can see dots. And so those dots have a certain sequence, and we can start reading the sequence from there. One big advantage of this technique is that it can be easily parallelized. You can have many strands of DNA on the same picture. But one big disadvantage is that it requires short fragments to be sequenced on each dot. So each one of these fragments cannot be very long, because then if this sequence gets too long, then in the fluid where it's swimming, more or less, it's going to start moving around and taking the place of other dots, and it's going to be really hard to tell apart. And this is different problem. It's an image segmentation problem where you're trying to isolate this dot, assign it to the same dot in the previous pictures, and know what the color of the sequence is. This is the second source of error, and this is what the sequencer will identify as high confidence dots or low confidence dots.